Hello everyone, I'm Chakrabarty, and in this presentation I'll go through the effects of modulating cyclic AMP with adenosine in efferent nerve terminals in the mouse bladder. ATP is a co-transmitter with acetylcholine at parasympathetic nerve terminals, and it has an excitatory role in generating nerve-mediated contractions in the detrusor from most animal bladders. ATP is released over lower stimulation frequencies, whilst acetylcholine release predominates at higher frequencies. However, in the human bladder, ATP only has a role in bladder pathologies, such as overactive bladder. Therefore, it would be highly desirable to selectively reduce ATP release from nerve terminals and leave acetylcholine release intact. In previous studies, adenosine reduces nerve-mediated contractions, with a larger effect at lower stimulation frequencies compared to higher frequencies. This action is mediated by adenosine A1 receptors, which reduces intracellular cyclic AMP and subsequently reduces activity of protein kinase A, PKA. In this study, we hypothesized that A1 receptor agonists do reduce nerve-mediated ATP, but not acetylcholine release, an action mediated by an A1 receptor cyclic AMP-PKA pathway. Intact mouse bladder tissue strips were used for conventional tension recording experiments, and the contractions were generated by electrical field stimulation. Tension was plotted as a function of stimulation frequency and analyzed to estimate parameters like maximum tension, Tmax, and the frequency to generate half maximal tension, F-half. A reduction in Tmax implies an action on force mainly by acetylcholine-dependent pathways, and an increase in F-half implies a force reduction mainly via ATP-dependent pathways. Nerve-mediated ATP and acetylcholine release was also measured from superfusate samples. Adenosine reduced nerve-mediated contractions at low frequencies, and you can see this at 2 and 8 Hz frequency in this figure, but had no effect at higher frequency contractions. When we look at parameters in a force-frequency relationship, we can see that there is an increase in the F-half, which corresponds with reduced ATP release, and that there is no effect on the maximum tension and acetylcholine release measured. The A1 receptor selective agonist, CPA, and the non-selective A1/A2 receptor agonist, NECA, had a similar result to adenosine on the F-half and ATP release. The effect of adenosine was blocked by an A1 receptor antagonist, DPCPX. These results are consistent with the hypothesis that adenosine acts via adenosine A1 receptor activation to result in a reduction in nerve-mediated ATP release and subsequent detrusor contraction. Activation of the A1 receptor results in the inhibition of cyclic AMP. Inhibiting cyclic AMP reduces PKA activity, and by directly inhibiting PKA using a PKA inhibitor also increased the F-half and reduced the amount of ATP that was released. Using agents that have the opposite effect, that is an increase in cyclic AMP, by using a adenylate cyclase activator like forskolin or cyclic AMP analog, had no effect on nerve-mediated contractions and ATP release. An additional target for cyclic AMP includes exchange protein directly activated by cyclic AMP, EPACs, and the addition of an EPAC activator or an EPAC inhibitor had no effect on nerve-mediated contractions or ATP release. This suggests that the main target for cyclic AMP in mediating nerve-mediated ATP release is via PKA and not by the EPAC route. So in this study, we've shown that adenosine selectively reduces ATP release from motor nerve supplying detrusor smooth muscle. The pathway of this action has been characterized initially by activation of an A1 receptor with downstream inhibition of adenylate cyclase, inhibition of cyclic AMP generation, and inhibition of PKA activation. Because ATP is associated with pathological contractile function in the human bladder, the ability to specifically attenuate its release by modulating cyclic nucleotide levels with cyclic nucleotides like cyclic AMP offers a novel therapeutic target. And I would like to thank everyone who has been involved in this project, funding bodies, the ICS for giving me the opportunity to present on this platform, and thank you to everyone here for your kind attention.